Assalamu alaikum. I am Zohra Naseem, Department of Zoology, Cohort University of Science and Technology, MPhil student. My presentation and research topic is molecular characterization of Babesia species based on cytochrome B gene in large ruminant of district Pajawar. Now start from the introduction. Babesiosis is a tick-borne disease of ruminant equine, cattle, and human brought by an intra-erythrocytic protozoan of the genus Babesia present worldwide but is most common in tropical and subtropical areas. Because Babesia prevalence is high in those areas who have a high temperature. Tropical and subtropical areas also have a high temperature, so that's why this genus Babesia is most common in tropical and subtropical areas. Large-scale mortality and morbidity are caused by it, particularly in developing nations like Pakistan. More than 100 species of Babesia are found around the world, but only two, Babesia bovis and Babesia begemana, are dominating in the tropical, subtropical, and temperate regions, affecting millions of livestock population. Pakistan livestock is also considered one of the most important commodity and ranked fourth in milk production worldwide. The current study is designed to find the actual genetic diversity of the Babesia species and to evaluate the parasite species diversity and evolutionary relationship in Khyber Pakhtunkhwa, Pakistan. My research objective to investigate the molecular diversity of Babesia species based on cytochrome B gene in large ruminant of district Pajawar, Khyber Pakhtunkhwa, Pakistan. Come to the material and method. Firstly, sample collection. Approximately 200 blood samples are collected by the help of a syringe from the jugular vein of large ruminant animal in district Pajawar on the basis of clinical signs and symptoms. Samples are stored in ADTA tube and labeled with the host name, month, gender, and age of the animal from which blood was taken, and then transported to molecular parasitology and virology laboratory in the Department of Zoology, Cohort University of Science and Technology. The collected blood samples were stored at -4 centigrade for further analysis. Then we done a microscopy. About 10 microliter drop of blood placed on a clean slide with the help of a pipette and fixed with a methanol. The Babesia slides were stained with Gamson stain and after a few minutes washed with the help of a distilled water. The slides were comparatively dry, then observed under high power 100x of microscope immersion oil was used, which condensed the light and make the slide more clear. This one is a microscopic picture of large ruminant blood. Then I done DNA extraction through Kygene kit, which have a 4-step lysis, washing, and the last one is elution. Extracted DNA kept -4 centigrade for further analysis. Then I done PCR polymerase chain reaction, which have 3 steps: denaturation, annealing, extension. Initial denaturation step of 5 minutes at 95 centigrade, denaturation for 45 seconds at 95 centigrade, annealing step of 35 seconds for 54 centigrade, extension phase of 72 centigrade for 45 seconds, and final extension 72 centigrade for 7 minutes. Then I done gel electrophoresis. 1.5% agarose gel is used. The positive PCR samples were sent to the commercial company for the sequencing to find the nearest related species of Babesia. The related match nucleotide sequence were selected. Multiple alignment was done with cluster W. Omega X was used for molecular evolution and phylogenetic analysis. This one is my study area map, which is Pajawar, the northern area of Khyber Pakhtunkhwa, Pakistan. Results: gender and month-wise prevalence of Babesia begemana. Female have a high prevalence as compared to male, due to the fact that they were kept longer for breeding and milk production purpose, support insufficient feed against their high demands. If we see a month-wise prevalence, July has a high prevalence rate as compared to other months because in July the temperature is high. Age-wise and host-wise prevalence, cattle have a high prevalence as compared to buffalo. If we see age-wise prevalence, adults have a high prevalence rate as compared to the young one. High prevalence were reported in animal aged more than 3 as compared to 1 to 3 age animal because in worse age resistance of the disease were adults show more susceptible than cause, protected by a cholesterol immunity. Area-wise prevalence of Babesia beggiana, Salarzai has a high prevalence rate as compared to the Balgina. This one is the microscopic picture and this one is the gel picture. Microscopic prevalence is high as compared to PCR because in microscopy we didn't know what it was, either it is a Babesia or Theileria. But in PCR, we confirm that this one is the Babesia and which species of Babesia is present, and we know the correct result. So that's why the PCR has low prevalence as compared to the microscopy prevalence. Phylogenetic tree. Phylogenetic tree represent evolutionary relationship among or between a group of organisms and show how species are evolved from a series of common ancestors. For example, Babesia bovis and Babesia ovata and Babesia beggiana. Both are distinct, but they have a common ancestor. So the phylogenetic tree helps or show the common ancestor. This one is the cytochrome B gene, phylogenetic tree of cytochrome B gene of Babesia species. Conclusion and recommendation: the highest prevalence rate of Babesia beggiana was found in female hosts, both cattle and buffalo. The cattle were more effective by Babesia beggiana as compared to buffalo. In month of July, the prevalence rate was observed high. In the middle age, 3 to 6 old cattle and buffaloes are more affected as compared to other lower and higher age group. Similarly, in Salarzai, the prevalence rate of Babesia beggiana was significantly high. The PCR was more sensitive as compared to microscopy. The prevalence measure followed by the owner of the cattle as well as by the government to minimize the prevalence ratio of the babesiosis. These are the references. Thank you. Allah Hafiz.